This video is going to discuss lysogenic replication. This is one of two ways that bacteriophage can replicate or increase the number of viruses present. So there's another video about the lytic cycle that you can watch to better understand that. So what we're going to start with is a bacteriophage and a host bacterial cell. In this picture, this red molecule represents the bacterial chromosome. And so the first thing that's going to happen, as we've discussed with all viruses, is they have to bind to, attach to, and infect a cell. So in this case, a piece of viral DNA has entered the host cell. So that's shown here in yellow. And it's at this point that a lysogenic bacteriophage can undergo one of two replication cycles. So again, lytic is one possibility, but this particular video is going to focus on the lysogenic cycle. So during the lysogenic cycle, what happens is the viral DNA actually integrates into the bacterial chromosome, and it's at this point that we now call this a prophage. So prophage is a term that we use to describe a lysogenic bacteriophage that has integrated into the bacterial DNA. And what then happens, as we've discussed many times, bacteria do something quite well, which is they divide by binary fission. So that one cell splits into two, and this continues through multiple rounds of binary fission. So two cells split into four, and this continues over and over again until you can have many, many descendant cells. And what's important to notice is that with each round of binary fission, the entire chromosome, including the viral DNA, is replicated. So every descendant cell that is descended from that original infected cell that has a prophage has viral DNA integrated into it. So every descendant cell has the viral genome present. At some point, what can happen is a process that's known as induction. And so what that means is that viral DNA can deassociate, can kind of cut itself out of the bacterial chromosome. And so now they're separate. And so again, that term is induction. And so this is a switch from the lysogenic cycle to the lytic cycle. And as we have discussed in the lytic cycle replication video, what's going to happen is this piece of DNA hasn't been used to make any viral parts yet. It integrated and was kind of riding the wave of binary fission, but no viral synthesis and no viral assembly was happening. But when there's a switch through induction to the lytic, this piece of viral DNA is going to start making viral proteins. And one of those destroys or chews up the bacterial chromosome, so it's no longer intact. Then the other bits of information here are going to synthesize all of these viral parts. And so there's the capsid, there's the tail fibers. And after those parts are made, that's viral synthesis, there is a process of assembling them. And so these should look pretty familiar to you because this is what a bacteriophage looks like. And so this continues until there's so many bacteriophage present that the final step in this process is the release of these bacteriophage. And that happens through, because we've switched from to the lytic cycle, the lysis of this bacterial cell. So this host cell dies, and upon doing so, the cell membrane loses its integrity, and these newly assembled bacteriophage are released. And you can then look at this, and this one bacteriophage can go find another host cell to repeat this process. So in summary, what we have is a host cell that gets infected by a bacteriophage. And right after that entry step, that's where there is this fork in the road, so to speak, where a lysogenic bacteriophage can carry out the lytic cycle, which is shown here on the bottom. And that's simply degrading the chromosome, synthesizing viral parts, assembling new bacteriophage, and then lysing. And then that's the lytic cycle. Alternatively, after entry, you can have the lysogenic cycle, which is, again, when that piece of viral DNA integrates into the bacterial cell chromosome. And then that one host cell copies its DNA along with the virus during the process of binary fission, and that cell splits into two. Each cell now has a copy of the viral DNA in the form of a prophage. It is integrated. Then those cells continue to divide over and over again. Induction is that process of switching from the lysogenic replication pathway to the lytic. And what that means is that the viral piece of DNA is going to start to synthesize viral parts, including some enzymes that start to cut up the chromosome, so the bacterial chromosome gets destroyed, and that piece of viral DNA is now separate from the rest of the bacterial DNA. And so that viral DNA can be used to, again, make viral parts, that synthesis, and then assemble those parts into infectious viral particles, the bacteriophage, and then the cell will lyse. And then each of those newly assembled bacteriophage can find another host cell and then inject their DNA in the form of the process of entering the host cell. And then that's again where either lysogeny or the lytic cycle can take place.